Thank you all for your perseverance, for your patience. This will take a little bit of a different direction, but it's consistent with what Gil said before about what digitization is, because DNA barcoding is a form of digitization. This is a little bit of a segue into tomorrow morning's session on DNA barcoding. I hope you'll attend that one as well. So we've been talking about different data types in the domain of biodiversity: specimens, things in drawers and bottles, taxonomic names for those things, characteristics, and I would say geolocation, georeference data, measurements, images, as well as DNA sequences are all characteristics. And finally, publications and aggregations of data in databases are all different types of information that can be analog or digital, frankly. And we've been talking about a little bit about ways to connect them. What are the linkages between them, not just electronic and informatics linkages, but logical and semantic linkages between them. The problem becomes really important as we scale up to global information resources about those things. So now we have the ability to really make large-scale errors in connecting things if we're imprecise or if we've designed our logical structure incorrectly. So most biological data, as several people have said, are indexed by names. So what's in a name? What's it used for? It's an indexing variable for retrieving information, and I could give a whole other talk about why Linnaean taxonomy is a terrible way to index information. It's also used as a vocabulary for basic research on evolution and ecology, so that we think we're talking about the same thing when we use the same Latin binomial. But we also use names to link as a linking variable between specimens in nature and other parts of civil society, like laws, regulations, quotas. Catch quotas, for example, in fisheries are linked to Linnaean names. So we believe then that legal action and market are all using the name in the same way. Protected status, inspections for pests and alien invasive species all use names, and we assume that we're all talking about the same thing and what we're talking about is clear. But in fact, that's often not the case, because the other thing that's in a name are varying levels of uncertainty. For a particular specimen, what is the correct name to apply? If that specimen, is that specimen a hybrid? What name do we put on a hybrid? If we're dealing with a tissue sample that doesn't display morphological diagnostic features, what name do we put on that tissue sample? DNA extracts, products, derivatives, all beg the question of what's the correct name for this thing if the name is linked to diagnostic characters, usually adult males, plants in flower, things that we don't always see. We don't always encounter organisms in that condition. So, I'll talk a little bit later on, we encounter things like dried meat in the marketplace or at border inspection stations, and we don't know if they come from primates. Sometimes they do. When we encounter eggs, we don't know whether, for example, this smuggler, who's got about 50 eggs attached to his torso, whether that's an endangered parrot. In this case, it was. We don't know if roots or seedlings that are encountered at border inspection stations are endangered species or noxious weeds. And when we encounter powders, we don't know if they come from things like rhinoceros horns. What else is in a name? Well, there's this confusion about the names themselves, and Shelley talked a little bit about it before under the global names user base, the relationship among all the variations on a name, synonyms, competing species concepts, different researchers that are using the same name for slightly different boundaries on morphological or other variation. Vernacular names, the association of a vernacular name to a Latin name introduces uncertainty. That's why DNA barcoding and other forms of DNA data, being digital in nature, can reduce some of that uncertainty. So the definition of a barcode, a DNA barcode, is a short gene sequence. It's taken from a standardized portion of the genome, and it's used to identify species. Well, more precisely, it's used to associate specimens with species names. So when you encounter fish in different forms, whether they're adults with diagnostic characters or larvae that are difficult to identify because they haven't developed diagnostic adult characters, or derivatives or products like fish fillets or caviar, they all bear the same DNA barcode if they belong to the same species, within a minor amount of variation. For animals, the barcode region is part of the cytochrome C oxidase 1 mitochondrial genome. It's about 650 bases long. And for other taxa, land plants, fungi, there have been approved barcode regions, 2 chloroplast coding regions for land plants, and a nuclear ITS region for fungi. Lower level of resolving ability for those in the 70 to 75% range, whereas CO1 for animals so far is over 95% reliable in diagnosing species boundaries. How does barcoding work? Most of the work in barcoding is in phase 1. I'll call it phase 1 barcoding, building a reference library. It's much like building an identification key. You amass information about the variability within a species and the distinctions between species, and you codify that in an identification key, which is extremely analog in nature. We're doing the same thing with DNA barcoding. You start with well-identified specimens that display all the diagnostic features that taxonomists rely on. Take a tissue subsample of that specimen, extract the DNA, amplify the barcode region, sequence that region, and then after a little bit of cleaning and checking, you submit that to GenBank or one of the other global databases of DNA sequences. And as that database becomes better populated, then it becomes more reliable for identifying unknowns. And you go through the same process. You take, for example, a rootlet like the one I illustrated a few slides back, or any fragment of a fish fillet. You subsample the tissue, extract the DNA, amplify the barcode region, and then sequence that, then compare it with the reference library, and that gives you a probability statement about what it is. So it's very much like an identification key, but on a much more digital and quantifiable basis. And here's where the data reside. They reside in GenBank, the European Nucleotide Archive, and the DNA Databank of Japan, which together make up the International Nucleotide Sequence Database Collaboration. They exchange data every night, so they contain the same data, and you can do the sequence, you can do the lookup in any of those. This is a schematic of the DNA barcode standard, or the data standard. Since we're dealing with standardized sequences, we have to regularize how the data are captured, how they're checked, what quality, what level of quality and completeness are required for a reliable identification. So the center of this is the barcode sequence. I'm only going to deal with a few of these linkages. The most important one is the linkage to a voucher specimen. This was one of the breakthroughs in barcoding, that very few of the sequences in GenBank were tied to the specimen, identified the specimen from which the sample and the sequence came, whereas barcoding requires that you identify the origin. And I'll go into that in greater depth. The trace files that come off the sequencer are required, the primer sequences that you use to do the PCR amplification, a species name that's identifiable, a whole bunch of other metadata about the locality, collector. All of those make up the data standard, but I'm not going to go into much detail. Here's what a GenBank record looks like. So you can see that this one, I should mention that the whole point of this is that data records submitted to GenBank or the others are checked for compliance, and if they comply, they get a reserved keyword barcode in uppercase letters. So here's a barcode record in GenBank. You can see the keyword barcode. It comes from the CO1 gene. Over here under links, there's a link to the trace file, so you can click on that and immediately go to the forward and reverse trace files, the raw data that came off the sequencer. And down here, there are links to the taxon name. That'll take you to a number of authoritative sources of information about that name. And I want to focus on this line, specimen underscore voucher equals UAM:MAM: 86887. If you click on that link, which is structured in this way, I'll get back to that in a minute, it takes you to the museum record, in this case the University of Alaska Museum, the record of the specimen that provided the tissue sample that generated the barcode sequence. So we've decided that as the world works towards the implementation of UUIDs, as Gil described them, we have a very workable system already, and it's the one that he alluded to, based on institutional IDs, 3 elements of the Darwin Core Standard: institutional ID, collection code, and catalog ID. So Gil started off by saying if you did a Google search on specimen 427, you get tens of thousands of hits. But if you do a search on NHM UK, which is the standardized ID for the Natural History Museum in South Kensington, London, :LEP, which is their Lepidoptera collection, :9427, together that's a unique identifier. If you concatenate those 3 elements, there's a very high probability that points to one and only one specimen. And it has the advantage of being in existence and in many cases digitized. It also links to the literature because specimens are usually identified in the literature using their repository and their catalog number. Too commonly they leave out the collection code. So if you did a search on NHM UK 427, you'd probably get 15. You'd get a fish, a mammal, a bird, an insect, a butterfly. But you could, if you knew that you were looking for a fish and you got the list of all the specimen 427s from London, you'd know which one you're interested in. And if you added fish in the middle for the collection code, you'd have probably a unique identifier. Now, I agree completely with Gil. For computers to communicate with each other, a UUID is much more efficient, much more stable. And linking these kinds of what we call Darwin Core triplets to UUIDs gives us a Rosetta Stone of going from the literature into databases. Now, there hasn't been, up until recently, a standard terminology for institutional IDs and collection codes. The botanists did the best job through Index Herbariorum. They have a very stable system of globally unique identifiers for institutions. And most herbaria don't have collections within them. They have a single numbering system. But when you mix them together and say with zoology museums or microbiological collections, you get a lot of collisions. You get a lot of ambiguous usages. So that's why we've created a new registry online called GR-Bio, the Global Registry of Biodiversity Repositories. It's going to be going online in the next few weeks. It is a registry of about 7,000 biorepositories, including all the contents of Index Herbariorum and about 3,000 non-herbarium institutions. And we're working out the ambiguities. There are now about only 129 ambiguous institutional IDs left, and we're going to be putting that list out on the web for crowdsourcing so people can say, oh, they stopped using that institutional ID long ago, or yes, these two still are in conflict. So we'll be able to resolve those. So there'll be an announcement probably by September that this is now live. You can go in, fill out information about your repository, update your record in Index Herbariorum. We hope that this will be the world's one-stop shopping for institutional identifiers. I want to talk about two ways that barcoding is done, two systems of digitization, if you will, and Bill talked about how and when do you digitize things with a goal in mind. And I'm just going to give two examples. The first is what I'll call collection-based barcoding. And this is the closest to the kind of digitization that big projects are doing, like scanning herbarium sheets. And this example comes from the U.S. National Museum at the Smithsonian Institution in Washington, where we decided that this was an extraordinary collection of frozen tissue that would yield a very large amount of high-quality data with a minimum of effort. The collection of frozen tissue includes about 20,000 specimens from 2,500 species. That's fully 1/4 of the world's avifauna. It's about 10,000 bird species that are known. 1/4 of them can be sampled in one place for their DNA. So what we did was we looked at which species had already been barcoded, picked out the samples that we needed to bring as many species as possible, up to 5 barcode records per species, and came out with a data set. We sampled about 3,200 tissues from the collection, so that's about 15% of the collection was sampled, representing 1,400 species. Some of those didn't generate sequences. The samples were degraded. They were still in an ultra-cold freezer, but they didn't have any DNA left in them. 94 of them had been badly mislabeled, probably mixed up in the field, and another 166 just clustered where they shouldn't have. So this turned out to be useful from a purely curatorial point of view. From a quality control point of view, we learned that some of the collection was not reliable. So now we can go back and sample again to weed out the tissues that no longer have DNA or aren't correctly labeled. That left about 2,700 what we call barcode-ready samples that we thought would meet the DNA barcodes data standard. 1,100 species were included in those. That means these were the first barcoded species, first samples to have these barcode samples. That's a 91% increase, almost a doubling of the number of bird species that had barcode records from one collection. So, very efficient way to do this. Here's the timeline for it. We began sampling tissues in the winter and spring of 2010 to 11, and the entire project was done. The data were released about a year later. And the total cost was about $25,000, about $10 per specimen. So this can be done very efficiently by mining tissue from the world's big frozen tissue collections. Another example takes a completely different strategy, and this is one that our organization, the Consortium for the Barcode of Life, is leading. It received a Global Impact Award from Google a few months ago, and it's called the Barcode of Wildlife Project. This is devoted to creating a barcode reference library of endangered species, CITES-listed species, that are the highest priority for 6 partner countries. The Google project has about $3 million in funding, and we're working with South Africa, Kenya, Nigeria, Mexico, Brazil, and Thailand. And each of those countries is going to be selecting about 300 or 400 species that they know are being poached or trafficked through their territory, and they want to investigate those cases and prosecute them, but they have problems identifying the species because they're being trafficked as either masqueraded or derived, where there are no diagnostic characters, or the very nature of the species, even when you get complete specimens, it's hard to tell them apart from close relatives. So this is highly focused on high priority species and their close relatives. The goals of the project are to demonstrate the utility of barcoding in investigations and courtroom prosecutions by the end of 2014. To do that, we're constructing a barcode reference library of about 10,000 species. 2,000 of them are the endangered species. 8,000 species, in addition to them, about a 4-to-1 ratio, are the close relatives or the lookalikes, anticipating the false negative and false positive problem, because we're going to be in court with defense attorneys that will say, we see that the barcode matches or nearly matches the record in the reference library. How do you know it's not something closely related? After all, there's a little bit of variability within the species. So we're preparing for that with this 4-to-1 ratio. We hope that some of these countries are going to see the utility of barcoding and take the next step and formally adopt it as a forensic tool and begin to support barcoding, not just forensic barcoding, but academic barcoding. In the next month or so, you're going to be seeing a call for participation by non-country scientists. We're not going to rely on the partner countries to provide the voucher specimens. We're going to be looking in all sorts of museums. We're going to be looking for taxonomic specialists who can help us identify what are the closely related species and the lookalikes we should barcode. Some of these are going to be taxonomic problems where we have genuine problems agreeing on what are the boundaries of these taxon concepts. Remember all of those sources of uncertainty I started describing? We'll need taxonomists to help solve those, and we have money in the grant to support taxonomic work to clarify the use of these names, as well as provide voucher specimens, as well as process samples in laboratories. So that's what we call an application-based barcoding as opposed to the collection-based barcoding, very parallel to the kinds of things that Gil was describing in the iDigBio project. So I'll stop there, remind you that there is another session tomorrow morning after the 2 plenary keynote talks. It's going to be in this room. Now we've mastered the technology of GoToWebinar. I hope there won't be any glitches. Thank you all for coming.
Are there any questions for David from the floor? Yes. Yeah.
This is a legal question. Do you know of any cases that have already brought criminals to justice based on DNA barcoding in the same species or countries that you have partnered?
There have been cases that didn't go to court. Most of the time so far, when the threat of DNA evidence is raised, people confess or they plea bargain. So there are several cases that are in investigation that are headed for court. Several in the United States. In the partner countries, there's one where a case is being built with barcode data. But these are going to be first efforts. I don't think there's ever been a full prosecution that had barcode data in it yet.
Early days.
Very early days. This is the equivalent of the early days of fingerprinting and human DNA identification. So it's a very exciting time. We're spending a lot of time talking to lawyers and prosecutors about what defense attorneys are going to attack. What kind of defense will they mount against this data? We're trying to anticipate that and build that into the project. So we had a very interesting workshop in South Africa several weeks ago that was half researchers and half police and prosecutors talking about how will this work.
Thank you.
Any questions online?
There are no questions online as yet. Anyone? Hang on. Here we go. This one. This is from Hector Aguilar. What can be done when intraspecific variation is larger than interspecific variation for a species?
We add a second gene region.
Okay.
Yeah, we take recourse in faster evolving gene regions, nuclear genes, for example, or a different portion of the mitochondria.
Okay, thank you. And he's followed up with, and if that is still not enough?
If that is still not enough, then then there's obviously taxonomy. You have to go back to the drawing board and figure out what is the taxonomy of this group. Are those really correctly done, correctly drawn species boundaries? At some point, you have to say, are these good names?
Yeah.